They are all curled up, and they have a lot of whitening. Normally, the infections happen in winter time for these nematodes, and also the growth. Then the new growth is challenged as well. We have also an important one in the grape growing areas of the state, the dagger nematode. This is an ectoparasite, so it doesn't infect the root and gets inside like the root knot nematode or the citrus nematode. But these nematodes, what they do is they have a very long stylet, like all plant parasitic nematodes have the mouth part, it's like a syringe, and they use it to pierce and get access to the plant tissues. So the picture here, kind of purple, shows the tip of a root, and you see a nematode there with the needle piercing the stem of the bottom of the root. And when they are doing that, actually the nematodes are injecting nipoviruses, and so they are vectors of these viruses that cause the yellowing on the leaves of the grapes. And you see yellow leaves when it's no fall, so there's not like there are fall colors. You see that in summertime and other times where the leaves should not be green and not yellow. Also, the leaves fan and curled up. That's why it's also called as the fan leaf nematode. Okay, so again, because we have become more aware of the challenge of chemical pesticides, scientists and people in the government and those ones that promote research have become aware of the impacts of chemical pesticides. So over the past decades, we have shown an increased interest for developing alternatives for pest control, not only of nematodes, but any other pathogens of plants. And one of the alternatives is looking for natural enemies or any other pathogens or byproducts of these pathogens that could potentially have benefit and ameliorate the effect of chemical pesticides. Nematicides are actually on the trend for enhancing their recognition and the ability to make sure that they are more promoted and explored as alternatives for the control of plant parasitic nematodes. This is very much actually true for European countries, which are actually on the top of promoting alternatives that are safe to the environment. So there's a lot of different alternatives that, again, natural products, natural oils, and also byproducts produced by microbes that potentially can be used for the control of plant parasitic nematodes. So with that consideration in mind, now we get into the good guys, and I will start talking about entomopathogenic nematodes. And I always tell my students, you get extra points if you can say all the name together without catching your breath. So entomopathogenic nematodes. And so those are beneficial nematodes. And again, when I say beneficial, it's from an anthropogenic perspective because it's beneficial to us humans, not to insects. So these are insect parasites, and they kill the when the insects are dead, you have here very interesting changes in colorations. In the scarab beetle above, you can even see that the worms are coming out of the insect. And basically, there are two major groups, ones that are called Steinernema nematodes and the other ones Heterorhabditis. And they have very interesting biology, and I've always been fascinated with them. I started working with nematodes since I was a PhD student, and from then on, they have been part of my life. And I always say, thanks to these worms, I'll travel around the world. So I'm thankful and devoted to entomopathogenic nematodes. So when I get chances like today to give a talk and educate people on what these nematodes are and what they do, I always get very excited. So I will review the life cycle because this actually will put us into context of what I'm going to be talking at the end of the talk, which is, and is actually the topic and the title of my presentation, about the bacterial symbionts of these nematodes. So you need to understand that these nematodes live in the soil, and they only have one free living stage that is called the infective juvenile, and it's actually, nematodes go through 4 juvenile stages in their life cycle, so they live more as juveniles than adults, like many invertebrates do. And so the third stage, infective juvenile, we normally call it IJ, to simplify, are the ones that live in the soil. And they can live in the soil for it. Probably no one really knows for how long, but for many, many, many months, and sometimes even years. And they can live there awaiting for the right insect host to come and parasitize. So when they are in that infective juvenile stage, these nematodes are a non-feeding stage. For those who are familiar with the research of C. elegans, how many of you have been familiar with C. elegans nematodes? Well, C. elegans nematodes, they also are free-living nematodes that live in the soil, and they have a, what is called in German, Dauer, which means resistance stage. And this Dauer stage is equivalent to the infective juvenile stage in entomopathogenic nematodes. So it's a stage where the nematodes are immature, so they don't reproduce, and they don't feed. Their mouth and anus are shut, so they don't require to feed. But that also causes some challenges, because having to live with whatever food they accumulated before they left the insect cadaver, that gives them the challenge that a limitation of their life. On top of that, there's another interesting component added to this story, that is that these nematodes don't work on their own. They work in collaboration with bacteria that they vector in their intestines. They are enterobacteria. And for Heterorhabditis nematodes, the bacteria are called Photorhabdus. A very interesting bacterium. It's the only bioluminescent bacterium in a terrestrial system, and is thought to have evolved from a marine environment. So the nematodes that brought the bacteria into the soil are thought to be marine nematodes, and the bacteria, if you look at the phylogeny at the molecular level, you see a lot of similarities with Vibrio bacteria that are also bioluminescent bacteria, Vibrio fischeri, and bobtail squids, and are also have a symbiotic relationship. So there are lots of interesting connections that bring these bacteria together. And Steinernema nematodes associate with Xenorhabdus bacteria. So again, for both groups, the bacteria live in the gut of the nematode. You can see, so the picture in pink there are in the infective juvenile nematodes, and the picture on the right is a close-up of the infective juvenile. And what you see in green inside is bacteria that were GFP, so labeled with a green fluorescent protein, and you can see the bacteria in the gut of the nematodes. So when the nematodes are in the soil, these nematodes move. They are very active seekers, and they are actually attracted by carbon dioxide produced by the insect host. They are also attracted to when insects that are herbivores, like a scarab beetle larvae, for example, feeding on the root system of a plant, that a wound that causes in the plant that produces the plants to secrete chemicals that are these volatile molecules that attract the nematodes. So there's a whole bodyguard hypothesis developed by a group of Dutch scientists that say that when plants are wounded, they produce these volatile chemicals that attract the natural enemies of the herbivores to be protected and prevent a further attack by these insects. So whatever the cues are, these nematodes find the host. They penetrate it. They can penetrate actively through piercing through the cuticle, or just getting through the body parts that have openings, like the mouth, the anus, or the spiracles. And when they go inside the insect, what they do is they release the bacterial symbionts. Some release them through the mouth, so it's a regurgitation, and that happens with the Heterorhabditis Photorhabdus partnership. And Steinernema defecate the bacteria, Xenorhabdus. So they have different strategies to release the bacteria inside the insect. So when that happens, that insect that at that moment was still alive, the bacteria produce an arsenal of toxins and secondary metabolites that kill the insect in a day or two. And the insect now becomes the food source for the nematodes and the bacteria that are growing and multiplying inside the insect cadaver. Now, very interestingly, and I'm going to bring this up in subsequent slides, the bacteria preserve the cadaver, and the cadaver doesn't putrefy for an extended period of time until the completion of this life cycle. So basically, the insect environment is this environment where these two organisms thrive and grow and multiply. So, and that's kind of like the payback of the bacteria to the nematodes that did them the favor of transporting them from one insect host to another. The bacteria don't live in the soil on their own. They rely on the nematodes to be moved from one insect host to another. So that's why we say it's a perfect mutualistic interaction, where the nematodes do the favor of transporting the bacteria, which are the true insect pathogens, into the insect, and the bacteria then, when they kill the insect, they preserve the cadaver in this protected environment that is not degraded, neither attacked by saprops in the soil. So when the life cycle is completed, the nematodes get, and this is actually thought to be driven by accumulation of ammonia by having so many worms there, and also food depletion. The infective juveniles, which are the grandparents, the grandchildren of the ones that infected the nematodes, because the nematodes, infective juveniles, went through multiple generations inside the insect cadaver. They leave the insect host and they remain in the soil, normally in packs, because nematodes, I always say, they are like wolves. They move in packs and they protect each other. And so that's the way they remain in the soil, awaiting for a new host. Now, these nematodes were discovered in the early actually in the late 1920s, 1927, 1929, by scientists in Brazil, where the first species was discovered, and then the next species was found in US in New Jersey. It's very rare to know the natural host of these nematodes, because in nature, and because the life cycle happens so fast, it's completed in a week or two, it's very hard to find insects infected by these nematodes. But there have been exceptions to the rules. And these have been cases where there are very active research projects on it in a given insect pest, and so natural infestations have happened. Or there have been cases, for example, in the case of crickets and mole crickets, when they a South American species infected Florida, researchers from Florida went to South America, actually Argentina, my home country, and Uruguay, to find natural enemies of these introduced crickets in US, and they found the entomopathogenic nematodes that were species from South America. But then, like I said, there's a wide range of species that from, lepidopterans, coleopterans, cicadas, even cicadas in Mexico, and that's actually, I'm bringing this up because it's one of the nematode species and bacterial symbionts that we are studying in my lab right now, and crickets and flies as well. So scientists immediately found the advantage of having these insect parasites and how they could mass produce them and formulate them as alternatives to chemical pesticides. And they have been under production and formulation for quite many decades now. There are companies in Europe, there are companies in US, and BASF right now is the major product company producing these entomopathogenic nematodes in US. There's a company in Germany called E-Nema that also produces these nematodes, and many other ones. And so they have done a lot of research in terms of extending the shelf life of the products and enhancing the formulations for reducing exposure to UV light, because normally nematodes, you find them in the soil. So how about controlling pests that are aerial? So for aerial pests, there are formulations that have these different alginate formulations and different clays that retard the exposure to UV, and they can be used in the gardens. Or in crops or greenhouses. The applications are numerous. There's a company in Tucson, Arbico, and those ones receive products. I believe they actually they receive the massive formulations from BASF, and then they rebrand them or repack them with their name. But they do have them, and they do sell them here for our community. And also an advantage is that any of the equipment that is used for chemical pesticides, it can be used for the application of these nematodes. So that is also a bonus added feature. Low scale applications in your backyard, or you could be doing applications of a full crop in a field. With the formulation enhancement, the host range of these nematodes has been increasingly expanded to many species, and like I said, from the field to gardens to greenhouses. In the mushroom production, Ciarius flies are a common problem for button mushroom production, and there are nematodes that are formulated. It's a species, Steinernema feltia, that controls very well fungal gnats. So you can use them there. The mushrooms are then cleaned, the nematodes are washed out, and they are okay for consumption. Lice and ticks have also been considered as targets for the application of these nematodes. And again, the possibilities are many, and people are, different companies are developing different alternatives in terms of formulations. And I would say constantly, I did this for many years. Early in my career, I was very much interested in understanding the diversity and distribution of these species worldwide. So I participated on a number of surveys in different parts of the world. And so there's always the interest for recovering new species, because it is thought that different species and strains could have different host range. And now, even with the consideration of the bacterial symbionts, that has opened the doors to increasing the surveys and the collection of novel species and strains. This slide is showing you a summary of the current commodities that these nematodes have. So you have the crops, the insects that are target, and the species that are used. These are not the commercial names. So this is the nematode species, either Steinernema or Heterorhabditis, that have been used and commercialized. So it is quite wide. And some species, actually some nematodes, have a very limited host range. Others are very generalist. They have a wide range of species of insects from different orders, but they are, so again, that is bringing up the concept that the discovery of new species opens the door for new targets in terms of pest control and other applications of these nematodes. So this slide is basically summarizing why these nematodes are beneficial and why actually there are advantages over chemical pesticides and other products. And again, one of the key features is the being very specific and having or a very limited host range. Some species have a wider host range than others, and that's why they need to be tested in the lab and in the field for non-target effects. So far, they have been found not harmful to humans and animals. They don't leave any toxic residues. They basically have no contamination or very little, if any, into the environment. have seen insects developing resistance, which happens with BT or other entomopathogens. There are many researchers have done studies looking at the compatibility between these nematodes and chemical pesticides, and some work very well. And so if you can get rid of the chemical pesticides, at least you reduce the amounts when you are doing the combined application of entomopathogenic nematodes and chemical pesticides. These nematodes, because they live in the soil, depending on the amount of the formulation that has been applied in the field and the field conditions, the environmental conditions as well, they can recycle in the soil. So once you apply it, most likely in the next year, you would have to use less amount than the one that you use in year number one. Again, easy to produce and formulate is one of the bonus added features. And there are many studies being conducted now as we have learned more about the genetics of the nematodes and their bacterial symbionts, that there are genetic manipulations that can be done to enhance the power of these nematodes in terms of speeding up their life cycle and making them more virulent. So there's a lot of potential, or crossing different symbionts with different nematode species. Actually, that work that we did a few years ago in my lab. So mentioned that these nematodes, when they kill insects, they don't do it on their own. In fact, they are just actually, they are, I always tell my students, is they are bus ride. Yeah, they are the bus rides of the bacterial symbionts, because the bacterial symbionts are the true insect killers. And in fact, a lot of researchers for many years have said, well, if the bacteria are the true insect killers, ditch the nematodes and use the bacteria, right? It's like another BT. Can we do that? And in fact, we cannot. And again, I have spent many decades of research in this topic, and also other people, because I told you that this is a very intimate type mutualistic interaction. When you disassociate, so when you can do research in the lab disassociating the bacterial symbionts from the nematodes, and they grow for a limited period of time in vitro and separately, but over time, either the nematodes die, they lose their fitness, or the bacteria also lose their virulence. So there's a very intimate association that we are still trying to figure out precisely what it is. We know for sure that the bacteria get a lot of nutrients from the nematodes when they are in that infective juvenile stage, dour stage in the soil. And the nematodes are nurturing the bacteria because they need them. The nematodes without the bacteria, yes, they can get into an insect, but they won't be able to reproduce, because remember, the bacteria are the ones that give the nematodes the right environment when they kill the insect to grow and multiply. So they need each other. In my lab, we have done a lot of research addressing the fitness cost of both the nematodes and the bacteria when they are separated, and then trying to associate them with different partners. And there are challenges in that association. Only in very limited cases we have enhanced both the virulence or the fitness of the nematode and the bacteria by doing different combinations. But the relationship is very tight. Each nematode species only associates with a given bacterial symbiont, and they would only carry that bacterial symbiont. Now, if you see the relationship from the bacteria side, there are certain bacteria species that are, I call them the promiscuous ones, because they associate with multiple nematode hosts. And there are evolutionary advantages for doing that, because in doing so, they have exploited a wider host range. And when I'm talking about host range, it's not only the nematode host range, but also the insect host range. Different nematodes have preference for different insect hosts. So the bacteria, by having different nematodes to vector them, enhance their perpetuation, their access to multiple insect hosts. So there are all these very interesting questions from an evolutionary perspective of these two organisms. So again, trying to find out how we can work with them and take advantage of them individually and do different options of looking at them from an alternative to chemical pesticides. So let's go back to the life cycle. And if you remember, I told you that the bacteria are very powerful in killing the insect, and they kill the insect in a day or two, something that a parasite would not do, you know. So when we are trying to define what these nematodes are from their lifestyle, I cannot say that they are true parasites, because a parasite will try to perpetuate the life of the host for as long as it can. Yes, it will debilitate it, but not kill it. So, but these nematodes, they need a dead body, right? So they are not really parasites, you know, they are feeding on bacteria that happen to be living in a dead body, and they vector it to that right host. So the definition of what these nematodes are is always very intriguing. No one has so far, in close to a century since research on this group of organisms was initiated, no one has been able to define, so they are not parasites, they are not parasitoids. Entomopathogenic, yes, but really actually not, because the pathogenic are the bacteria, not the nematodes, so they are vectors only. And if we look at their feeding habits, they are bacterivores because they feed on bacteria. So they are a very specialized group of bacterivore nematodes, not like the free living, but they have evolved to live in a very restrained environment, which is an insect cadaver. So again, the bacteria do the nasty job. They kill the insect very fast. And I told you that they do that by the release of a number of toxins. And different Photorhabdus and Xenorhabdus species, they have different amounts of toxins and genes and gene clusters that are involved in the production of these toxins. And they also are involved in the production of secondary metabolites. And these secondary metabolites are the ones that also preserve the cadaver from decomposition, because if decomposition happens, the life cycle of both the nematodes and the bacteria will be terminated, and they would not be able to perpetuate each other. So the cadaver needs to stay intact for as long as you can. And actually, when we do infections in the lab, if you look at the cadaver, not only it changes colors, like you see the one up there that's red, that's a scarab beetle larvae infected with Heterorhabditis Photorhabdus partnership, but they don't smell. They only start smelling putrid at the end of the life cycle, when most of the nematodes have already come out of the insect. And then you know it's time to just stop collecting the nematodes, because they come on their own. And we collect them in these petri dishes that we put a petri dish where we have the cadavers, and then we put that petri dish inside another one that has water. So basically the petri dish with the insect is floating. And then you see the nematodes abandoning the cadaver. The nematodes move, they go up to the petri dish, and they jump onto the second petri dish where it has the water. So they are very active, and they search for water like all nematodes do. They have positive hygrotropism. But that's the way we collect the nematodes without having to do a dissection or getting into any nasty or dirty job. So when I'm talking about preserving the cadaver, I always tell my students, this is like the US Enterprise in Star Trek, you know, when they put that beam of shield, you know, protecting the Enterprise from the Klingons' attack, right? So that's exactly what the cadaver is preserved. Like I said, it doesn't smell, there's no putrefaction, and no other microbes in the soil, which are many and plentiful in the soil, attack them. Also scavengers like ants. Ants, actually the Photorhabdus produce an ant deterrent factor that repels the ants from getting close to the cadavers that are infected by these nematodes. So very powerful chemicals in that insect cadaver being produced to preserve the cadaver. And a lot of research has been done, and people have found that at least there are 4 applications so far that have been identified as compounds that are involved in antibacterial, antimycotic, insecticidal, and also nematicidal. And I mentioned before that if looking at the genomes of these bacteria, and specifically in Photorhabdus, and that's where I'm diverting the rest of my talk, focusing on Photorhabdus. About 6% of the genomes of these bacteria have gene clusters that are involved in secondary metabolism, and also in the production of a number of toxins. These are some of the chemicals that have been identified, widely studied by a group in Germany and also in Canada. John Webster at University of British Columbia was one of the pioneers. He even also found out that some of these molecules have a cytotoxic effect. So there was a lot of interest, and I don't know, because this is all top secret research from a pharmaceutical perspective, in perhaps testing some of these products as anti-cancer drugs. It's a long shot. Research in an academic lab to do that, but so I'm focusing on other areas. And so what I have been working on over, I would say probably the past decade, is the study of this Photorhabdus bacterium that is subspecies sonorensis, and as the name suggests, it was found in the Sonoran Desert. Firstly, not on our US side, but actually in the Mexican side. It was found by a former graduate student that I had, and that collected cicadas in asparagus fields. He was doing a project in asparagus and found that infected cicadas with this red coloration. And then when we allowed them to complete the life cycle, we saw the nematodes coming out, and so that's when we discovered this species. The picture that you see there is the bacteria go into two different phases, phase 1 and phase 2. And so depending on the phase that they are, we grow them in an agar that's called NBTA for it's a bromothymol blue agar, and so the bacteria in phase 1 absorb that red coloration from the agar medium, and we have been working on them for looking at what activities these bacteria have to see if we find something novel or unique. So for quite a few years, I had a master student who actually first, he started looking at crude extract activities, such as getting the field rates of these bacteria and looking at the different activities. And we found that the 4 different kinds of activities that people have found, we also found them here. What you see here is different strains of Photorhabdus, except for TSB, which is just the control. And we saw that in both cases, and especially the red and the green, which are the strains from Arizona, show high levels of insect mortality. We tested at that time in corn earworm larvae, and it seemed to be very successful, at least killing 50% of the population at the concentration of 4 micrograms per, actually microliter should be in that slide. And the same with insect mortality, looking at infective juveniles. We also look at antimicrobial activity, both for antibacterial and antimycotic. And we also saw that both the Caborca and the CH35 strain had promise for activity against Fusarium oxysporum, a very ubiquitous fungus, and also Pseudomonas syringae, very important plant path. So with this evaluation of crude, this student graduated, only did a master's degree, but he opened the door for future work, which is actually the work that one of my current PhD students is doing. And we wanted to really look at the activity of specific compounds. Excuse me, I'm still recovering from a bad cold that I have. And we looked at different molecules because of, we have a patent disclosure, and I cannot reveal the names of the compounds, but those compounds that we call them as AK1, 2, and 3 have names. And we have identify them first through the isolation of these compounds through HPLC and then mass spectrometry. And so we started looking at the activities of these compounds. Because root knot nematode is a very nasty plant pathogen, we decided to cover first the research on this plant pathogen. And the targets that we are having is looking at the infective juveniles. So that's the stage that infects the roots. These are the plants, and you can see this is tomato seedling. And you can see the knots or knobs. And so we look at the lethal concentrations of each of these three compounds, and we also try to assess their effect on non-target nematode species against nematodes that are beneficial, free-living bacterivores, and also entomopathogenic nematodes that we don't want to kill if they are in the soil. So this slide looks at the results of what we did for the first compound, AK1, and as you can see, it has a nematicidal activity against the root knot nematodes at the concentration more or less at 67 micrograms per microliter. We start seeing a mortality of infective juveniles. And so interestingly, in examining all these compounds, we had, and I will show this towards the end, a whole range from true killing of the nematodes through inducing paralysis that sometimes reverts to going back to the activity. So this is what we have been looking at. But at least for AK1, we were very happy to see that there were no effects on nematodes that we didn't want to kill, which are, again, C. elegans is a free-living bacterivore nematode, and also entomopathogenic nematode species. There was a correlation between different timing of the exposure of these nematodes, the root knot, at between 24 and 72 hours. And we can see that as the time progresses, the mortality also increases relative to that. AK2 is the second compound, and we also had similar activities with very little effect or no effect on non-target species, but exhibiting mortality. And actually, AK2, if you remember from the previous one, AK1 had a concentration, a lethal concentration 50 of 67 micrograms per milliliter. This is much less, so it seems to be a more powerful compound. And there was not much difference in terms of the exposure time and an increased mortality for this particular compound. And the last compound that we have is actually a compound that was very interesting to us because we saw not much of mortality, only when we had very high concentrations, which is extremely high if you were to extrapolate that into a field application, 300 to 400 micrograms per milliliter, and also affecting C. elegans, which is not a good thing. But what we observe is that there's an induced quiescence. So the nematodes get immobilized, but then after you wash them out of the exposure of this compound, or you extend it over time, these nematodes reverse back to being active. So the question would be, if this is a valuable consideration of this specific compound for the testing of our application into plant parasitic nematodes. Right now, we don't know. Like I said, these are all laboratory assays. These are all in vitro assays. These are not in planta. So our next step is to move and do infections in the plant and see if we can have plants that have been exposed to the nematodes and see if we see a reduction in the formation of those knots, which means that the nematodes have parasitized the plant, and see if we can assess that, and that's what my student is going to be doing next semester. So in summary, so we can say that both AK1 and AK2 have promise for their nematicidal activity. You can see this graph here that shows green is when the nematodes are alive. So we have tested concentrations of 40 or less, and the nematodes, at least with AK1, remain alive. And only above 40 micrograms per milliliter, they become nematicidal. And you can see that and compare it with the different species that we tried, again, all non-targeted species. Again, we see that there is a lot of promise here, at least for 2 of the compounds that we have tested. And we are embarking into moving into at least greenhouse experiments to look at the activity of the 3 compounds in relation to plant parasitism. Also not listed here, but it is in my acknowledgements. Another student, Luis Ruiz, he's working on the antimycotic activity of these compounds. And so we have a wide range of fungal species that we are testing, all plant pathogens, and we actually are seeing also very promising results. So with that, I guess I will conclude my talk. Again, thanks to Ayako, who's my PhD student working on the root knot nematode, Luis, and other members of the lab, and of course, funding agencies, and also my colleague Isvan Molnar, because all the HPLC and SM analysis was done in his laboratory, and he's one of our collaborators. So I'm open to questions, and thank you for your attention.
Thank you very much, Patricia, for this, what I think is an incredibly complicated, interesting, and scary seminars. I never would have thought that under feet we have this world that has all these interesting, bizarre properties. Questions? Yes, sir. Yes.
This is truly fascinating, and much of the soil in this area is heavily compacted and would benefit by composting effectively, but one of the problems with composting is an issue of rodents and other things that can get into them, so there's a social stigma to. Now, is there a special formula of wonderful, healthy nematodes that could be part of an aliquot put into compost and overcome some of these issues that are part of a unhealthy ecosystem that you could convert into a healthy ecosystem?
You know, actually, that's not in the league of my work, but I bet you're talking about decomposting soil, right? Yeah, like I said, nematodes, you know, are involved in nutrient recycling, so they do it in nature at mini scale, right? And you see a lot of these bacterial-borne nematodes when you have compost. When I lived in California, that I would have the opportunity to have a little vegetable garden and grow vegetables, I would go and collect the soil there, and it was amazing the amount of nematodes we would find there. But someone massively doing that, I don't know. I guess they're leaving that to the earthworms only.
About turning bodies into soil.
Mm-hmm.
Anyone read that?
No.
Is there a methodology that you happen to know of that will instantly, or in several days, apparently they said it's a very quick thing. Would the nematodes be part of this process, do you think?
I bet they would be taking advantage. I mean, especially all the microherbivores, they would be there. How much impact they would do and help in the process, I honestly don't know. But it would be something interesting to explore. Yeah.
When you talk about the secondary metabolites, I don't think you mentioned what the original source requirement is to isolate a given amount of the metabolites. Is that scalable, or is it in such minute concentrations that
it can be scaled. And this is something I'm glad that you're bringing this up, because it's very interesting. So, scientists, we always try to answer the questions, and when we do that, sometimes we kind of narrow or simplify our arena or experiment just to get the answers that we want. So I told you that you can disassociate the nematodes and the bacteria, and we can grow the bacteria in a petri dish. Now, the compounds that are produced in the petri dish are not the same if you were to grow the bacteria in the insect. And no one has looked at what happens when they are in the nematode side. So, but it's known that different compounds are produced depending on where they are. The in vitro is a totally artificial situation. And, it's already known that some metabolites are not produced in vitro, but they are only produced in vivo. So the question is how to maintain an in vivo production massively to get these compounds, and identify them. And some have so I chose 3 products, 3 compounds, but my student found about 16. And some have very low peaks, and we cannot even try to work with that, because the amount of cultures that we need to produce and, get an amount that is reasonable, it's basically impossible, at least in our, right?
But do you think now that the identity of at least some of these compounds are known, it should be able to be synthesized?
Oh, yes. And that's the next step. Well, actually, one of the compounds that we have is already a molecule that is known. So, but it was never tested for this purpose. So, like I said, having it commercially available, yeah, it cost, I think, $300 for 100 micrograms, something like that. And so, yeah, but you can do it.
So the secondary metabolites are coming from the bacteria?
From the bacteria.
The one that causes quiescence, do you think that's some kind of signaling between the bacteria and the host to maybe like, this is the time to abandon the food source, or this is the time to...
So yeah, that's different, because the quiescence is on a plant parasitic nematode. So not all nematodes are associated with bacteria, and not all the associations are the same. So like I said, there are nematodes that feed on bacteria. There are nematodes that vector bacteria, like the ones that criconematids that transmit a number of fungal diseases or bacterial diseases into plants, and the entomopathogenic that, like I said, happen to vector it from one insect to another. But yeah, honestly, I don't know. With the connection with the plant parasitic nematodes, I don't know, because apparently they don't have bacterial symbionts. Now the concept of bacterial symbionts and symbionts of the nematodes, I was totally unknown for many years. Since people started looking at this system, they started finding out that there are more bacteria associations with nematodes than one would have imagined. In fact, a lot of the marine nematodes, which happen to be the most diverse group of nematodes in the planet, yet the most unstudied. There were pictures from last century that nematologists drew, and they were beautiful nematodes that look with hair-like structures. Well, those now are known that are ectosymbionts. They are bacteria that grow on the surface of the nematodes, and they are important to help the nematodes live in sulfur oxidizing environments like thermal vents. So, yeah.
The different species of bacteria that can go into a nematode, do they fight for dominance inside the host, or
no?
Can you
No, it's
multiple?
it's a very selective process. You can have 2 bacteria cultures, and the nematodes will only get 1. And now the bacteriologists say that there's colonization. The nematologists will say there's selection by the nematodes, choosing which bacteria. So it's like depending on where you see it. Yeah.
You described very eloquently the relationship between the nematodes, the bacteria, and then the prey that they have on insects. It sounds like the nematodes are now the cockroaches of the invertebrates. What preys on the nematodes? Do they have enemies?
Oh yes, they do. They do. Well, the nematodes live in the soil, so no, because they are unseen. So so you can find them 30 centimeters underground. Some species go deeper, others go more superficially. Some actually seek for their hosts if they were snakes. Others, they sit and wait, and when the insect passes, they jump. Yeah, well, they have nematode trapping fungi. So nematode trapping fungi are very ubiquitous in many soils, and so they don't segregate or discriminate what nematodes they get, whatever they got. But that's actually one of the potential challenges. Mites are the archie enemies of any nematodes. If I get a mite infestation in my lab, I'm doomed. And I had it before. I lost a lot of cultures. Yeah, just the dust mites. Yeah.
So they are part of the food chain.
Oh yes, they are. Yeah.
You mentioned an interface with viruses, and if we look at one of the human health issues of drug-resistant bacteria being killers, is it possible that we should be including viruses and the nematodes in a more complex ecosystem to either come up with new molecules or a new methodology of finding bactericidal materials?
Mm-hmm. Oh yeah, definitely. Yeah, like I said, all these molecules. I have another person in my lab that she's looking at human, pathogens, and so yeah, the possibilities are many. So the bacterial symbionts are considered a gold mine for discovery of new molecules. Mm-hmm. But you need to come and look at them getting the nematodes, because you can't find the bacteria alone. So they come in a package. And for now, until we understand what they give each other, you need to have them together. Because a lot of labs that work on bacteria, they lose the nematodes because they don't care about the nematodes. And then the bacteria, yes, they work for a while and everything is fine. And then they don't have it. And then that's when I get a phone call. Patricia, do you happen to have this nematode? We lost the host. We kept the bacteria because it's a bacteriology lab and they don't care about the nematodes. And I say, well, yeah, sometimes I do, sometimes I don't. Yeah. Yes.
I was curious as to why the nematodes produce an ant deterrent. Are ants like a predator for them?
Yes, so yeah, they do it to protect the cadaver, so the cadaver is not affected.
So they wouldn't attack the ants like they attack the cadavers?
Yes, they just don't go there at all.
Okay.
Yeah. And something actually is not, well, kind of related to that, but no one has had the answer yet. So I told you that Photorhabdus is the only bioluminescent bacteria in the terrestrial system. They have the lax genes that other bioluminescent bacteria have. And no one has found out what is the advantage of glowing in the dark is, if it's an attraction or it's a deterrent factor.
Anybody else? If not, thank you again.
Thank you.
For a very interesting and challenging presentation.
Thank you.
Much appreciated.
Thank you for having me here.